Okay, good morning, everyone. So thank you all for joining us this morning. We have a special guest with us this morning. So I'd like to introduce Dr. Nicholas Gonzalez Castro. He's an assistant professor of neurology at Harvard Medical School and neuro-oncologist at the Center for Neuro-Oncology, Dana-Farber Cancer Institute, an associate neurologist at the Brigham and Women's Hospital Department of Neurology, where you're also vice chair or associate vice chair for faculty wellness. He obtained his MD PhD from Harvard University, and his PhD research was in systems neuroscience, where he dissected motor learning mechanisms in humans aimed at improving rehabilitation protocols for stroke patients. He went on to complete his neurology residency and master at Brigham, followed by neuro-oncology fellowship at MGH and the Dana-Farber Cancer Institute. He received an NCI R25 grant while in residency to study oligodendroglioma heterogeneity, followed by a to characterize the landscape of chromatin accessibility within cellular states of glioblastoma. And then in 2020, Dr. Gonzalez Castro joined the faculty at QWH and the Dana-Farber Cancer Institute. So I could go on to describe more of his research, but I believe that's what you're going to be talking with us this morning. So thank you very much for joining us. And I
Good morning, everyone. Can you hear me online as well? Good. Thank you so much for the introduction, Dr. Sherman, and thank you to you and to the team for such a warm welcome of hospitality. It's very much appreciated. I would like to take a few minutes today to tell you a little bit about my work and what I think is my philosophy, or the philosophy that I'm trying to develop to try to achieve a dual goal of trying to provide better care for patients while at the same time advancing our understanding of the disease. These are my disclosures. They're not relevant to the contents of the presentation. I'm just going to. So I will make a claim, which is not a new claim, that medicine begins at the bedside. And this is a tale of two tumors. And these are the tumors that we're going to be discussing. I was told that this is a teaching conference, so you might want to guess what tumors we're going to talk about. And these are my patients, so they're real people. If medicine begins at the bedside, clinical observation is the foundation of medical science. And this is also not
recording in progress.
I'd like to bring you back in time and talk about Hippocrates of Cos, who recognizes the first physician that we have any knowledge of in Western medicine, and a text that he wrote called "On the Sacred Disease." And you get to play a game and guess what disease we're going to be talking about. But this is a disease that back in those days was thought to be related to afflictions given by the gods to individuals. So individuals were struck by the will of the gods, and they suffered from this, or they were possessed by spirits or demons. And Hippocrates, through keen observation, he says, but note, the cause of this malady is the brain, as it is in other serious diseases. And then he tries to provide a mechanistic understanding of this disease. And as you will see, he doesn't get it right. He relies on ideas from his time. But look how close he gets by explaining particularly. If, however, the flow of phlegm has been blocked from these pathways and it goes into the vessel going into the brain, which I have described before, it will create a congestion and the person becomes speechless. My patient, the one whose MRI you saw left, also became speechless when he first. And he gags. Froth flows from the mouth, the teeth are clenched, and the hands are contracted. His eyes are deviated, and he loses consciousness. So this is 2,400 years ago. just with a pair of eyes and a keen brain to think about this problem. So the disease that we're talking about, of course, is a seizure, and possibly, given the life expectancy of those times, it could have been a patient with the disease that we're going to be talking about. Let me bring you closer in time. Is there a way I can make this? And these two men, Harvey Cushing, the father of modern neurosurgery, trained at Hopkins with Halsted, and then called in to be the initial chair of the Department of Surgery as Peter Van Brigham, the precursor of the Brigham and Women's Hospital. And then Percival Bailey, who was his resident and graduate student, and together, along with Louis Eisenhardt, important to recognize the role of women in science as well. They put together in 1926 what I think is the first modern classification of primary brain tumors. And as you can see there, a classification of the tumors of the glioma group on a histogenetic basis with a correlated study of prognosis. You know, histogenetics has nothing to do with genetics, because in 1926, the concept of the gene and the idea of chromosomes, was starting to emerge. But we didn't even know, exactly how genetic information was transferred. It has to do with histogenesis, with looking under the microscope and observing different tissues. And then in table 4 of this book, they list a table of tumors, and they write on these columns the expected prognosis and the known survival. And I bring your attention here to tumor number 3, a tumor which back then was known as spongioblastoma multiforme. And you see that if you look at column E, the average survival in months from earliest known symptoms was 13 months, 1926. However, at the bottom of the table, we see another group of tumors, including oligodendroglioma and different astrocytomas with slightly different histology, where patients live 4, 5, and plus years after the onset of symptoms. In 1926, Cushing and Bailey already knew about IDH mutant glioma, which is the first tumor that I'm going to tell you about. And then we come now to the 21st century and the work of Bert Vogelstein and other colleagues at Hopkins, where they start profiling glioblastoma, one of the first tumors of which we started to look at genomic information. And they observed that those tumors that have a mutation in isocitrate dehydrogenase have a much better prognosis than those that are identified for this gene. And this is later shown in a tumor back then was known as anaplastic astrocytoma. Same phenomenon here. So in the last 10 years, we have gained a lot of understanding, and we have now the current classification, the 2021 classification of diffuse infiltrating gliomas, which relies heavily on the presence of IDH mutations to really be able to tell apart these two classes of tumors, along with other molecular alteration. But presence of an IDH mutation is a truncal phenomenon that separates these tumors distinctly. More recently, in the last 10, 12 years, through advances in microfluidics, in chemistry, in bioinformatics, we've been able to leverage a number of technologies to really be able to characterize tumors at single cell resolution. Because the cell, which is the basic unit of life, is also the basic unit of cancer. And we need to know cancer at the cellular level to really understand it completely. Leveraging these techniques, we have been able to characterize that gliomas, which sometimes look homogeneous under the microscope, have remarkable heterogeneity that goes beyond specific genomic alterations and can be observed in terms of gene expression profiles. And this is true for glioblastoma, for IDH mutant glioma, as well as for other gliomas. So let's go back to my patient, which when I first met him, he was 26, and he had presented after an episode of speech arrest, which we know was due to a seizure, and was found to have that insular lesion with T2 hyperintensity and with a slight decrement in flare. I'm not showing you the T2, but it's the T2 flare mismatch, which is a predictive sign of an IDH mutant astrocytoma, which is what this patient has. An IDH mutant glioma is a tumor of young people, of individuals in their 20s, their 30s, their 40s. And it comes in two main histologies, astrocytoma and oligodendroglioma. And oligodendroglioma tends to affect folks that are a little bit older, in their late 30s, 40s, sometimes 50s. I have a patient that is 64 with the recent diagnosis of IDH mutant glioma. Oligodendroglioma probably has had it for a long time. And the initial event that leads to oncogenesis is related to a mutation in isocitrate dehydrogenase. IDH is an enzyme involved in cellular metabolism, one of the enzymes of the tricarboxylic acid cycle that catalyzes the conversion of isocitrate into alpha-ketoglutarate. However, when the enzyme is mutated, alpha-ketoglutarate is taken as a substrate and leads to the production of the oncometabolite 2-hydroxyglutarate. In normal cellular physiology, there's a mitochondrial and cytosolic IDH, and through the production of alpha-ketoglutarate, which serves as a cofactor to a family of demethylation enzymes, like the TIP family and others. This leads to DNA demethylation and maintenance of chromatin structure in normal cells. But in cells with a mutation, the absence of alpha-ketoglutarate and the activity of 2-HE itself leads to inhibition of these demethylating enzymes and hypermethylated phenotype that leads to alteration in chromatin structure. And this is a key oncogenic phenomenon that leads to the interaction of oncogenes or proto-oncogenes with enhancers that are from unrelated genes and expression of oncogenes. For example, PDGFR8 is a beautiful study by Will Flabahan and Brett Bernstein in 2014 that showed increased interaction between an enhancer that was unrelated and PDGFR8 and increased PDGFR8 expression, and also more recently the beautiful work published by Dr. Jenkins here at Mayo, highlighting a specific SNP on chromosome 8 that is associated with disruption of OCK binding and increased expression of another oncogene. Leveraging single-cell profiling methods, we have observed that IDH mutant gliomas at the transcriptional level are composed of different cell states. Some cell states that resemble stem cells, called stem-like, and some cell states that are more in line with differentiated cells, such as oligodendrocytes or astrocytes. And this cellular state hierarchy, malignant cellular state hierarchy, resembles the normal hierarchy of cell types that we see during development, originating from a neuroprogenitor cell, an NPC, into subsequently more differentiated cells. This was work done by Mario Suva and Tati Roche. One of the key problems with IDH is that although these tumors are slow-growing, ingramescent initially, over time, over a period of a decade or so, they invariably transform into a very aggressive tumors. And since we know about the heterogeneity of glioma, one natural question is, what cellular states are associated with progression in IDH glioma? And to study this question, we generated single-cell RNA-seq and single-cell attack-seq data from a number of patients at our institutions, And for big questions, you need big data, and we partnered with a number of institutions here in North America and in China, and we leverage a large data set to answer this question. In addition, we took advantage of recent data sets profiling normal developing brain and adult brain at single cell resolution, both by RNA-seq and ATAC. And we took our malignant cells and projected them over the clusters of normal cells, showing a significant overlap, as I was highlighting in the previous slide, that these are cells that are malignant, but they're very much related to normal counterparts during development. And here, on this study, which uses a slightly different technique than the initial study done by we defined one additional cellular state, the GPC, glial progenitor cell-like state. So with improvements in different methods, there's additional refinement of different observations that we have.
Nick, yeah. Quick question, and can they hear us? Oh, yes. Quick question, just if you don't mind, how many patients are we talking about here among all these centers for the specifically these are IDH mutants, is that correct?
IDH mutants of different grades.
Of different grades.
Yes. And here we had 55 samples in total for RNA-seq, and then we generated ATAC-seq data for 10 patients.
So 65 patients total among all these institutions. Okay. These are unique patients. And the tissue was just bone tissue, no specific location, margin, core, frontal lobe, temporal lobe, nothing like that.
Yeah, so for most of them, we don't have that annotation.
Gotcha.
And, we know that it was lesional tissue,
Gotcha.
but we don't know exactly where.
That's okay. That's how I collected my tissue for 17 years. But the last 5 years I have modified this tissue collection routine. We can tell you more about this.
That's a very important point. And when I discuss the other tumor now, I'll make some observations in that regard. So then we center our attention just on the malignant cells. And we saw, clear clustering at the RNA-seq level along these 4 different cellular states. And very much like we see in the normal cells, there's a good cell-to-cell correlation between cells and cellular states. So like an AC-like cell is most alike like an AC-like cell in RNA-seq. But on the ATAC-seq space, we observed that this correlation was disrupted in malignant cells. So although the correlation in chromatin accessibility space is preserved for normal cells, for malignant cells is a lot more diffuse. So the idea here is that in normal brain, cellular types are separated by high epigenetic barriers that keep cells being what they are. Well, in the malignant cell states, the epigenetic barrier is lower, enabling transition or plasticity between cellular states. And if there's transition or plasticity between cellular states, the question is, what is the cellular state compartment that is expanding as tumors progress in terms of grade?
And do you think that is directly due to the epigenetic modifications from the 2HG?
That's a good question. I think it's a cumulative effect over time. And I think that the elevated levels of 2HG and the disruption of a lot of regulatory elements of chromatin structure early on leads to subsequent events that just makes this a lot more permissive. But the specific details we don't understand yet. So we tried to look for what cellular state was expanding as the tumor went from grade 2 to 3 to 4, and then normal candidates stem-like states, the OPC like state, and we didn't see an increase there, but we did see a clear increase in the MBC cellular state as tumors progress in grade. And also looking at cycling genes, we see that the MBC cells are the ones that are cycling and proliferating. And lastly, we looked at the effect of the size of the MBC compartment with prognosis, and we see that those patients that have an expanded MBC compartment do worse. In addition, looking at copy number alterations, we see that as the tumors get enriched by these MPC cells, there's a greater accumulation of alterations in oncogenes and loss of tumor suppressor genes. And we see that this is a clonal effect. This is an example for one of our tumors where we see that an initiating event that is seen in 3 different subclones is gain of cyclin B2, followed subsequently by loss of CDKN2A, a tumor suppressor, and then lastly, MYC-N amplification. And when this last step takes place, then we see real enrichment for the MPC cells. On August 6, 2024, the FDA gave approval to vorasidenib, an oral IDH1/2 inhibitor that is now approved for the treatment of low-grade IDH glioma. So we've had great interest in studying the IDH inhibitors and their effect on tumors. And in fact, for both vorasidenib and an earlier compound, ivosidenib, which is an IDH1 inhibitor, we have been collecting samples of patients treated, and we observed that in patients that were treated with the IDH inhibitor, in panel A, in unmatched samples, we saw an expansion of the AC compartment, so the more differentiated compartment. And we also observed the same thing in a patient for which we had pre and post treatment tissue. And then doing a general analysis on our samples, we see that there is increase in the differentiated cell state. So very much IDH mutations are present in other tumors, such as in leukemias and cholangiocarcinoma, and they have been approved for those indications as well. And just like in leukemia, treatment with an IDH inhibitor leads to the removal of differentiated block. We see that same effect in our tumors when they're treated with an IDH inhibitor. But since the IDH inhibitor is essentially also a demethylating agent, we wonder what are additional mechanisms of demethylating agents and IDH inhibitors that prevent progression in IDH. So we went back to our data, and one thing that we observed first, mining data from the Cancer Genome Atlas, is that there was a significant increase in interference signal in wild-type tumors in the TCGA versus mutant. And we also see that looking at our own data, our single-cell RNA-seq data, we see that in mutated tumors, interference signal is much lower. So we conducted this experiment where we generated patient organoids. This is fresh tissue obtained from patients that is dissected, and essentially these are large clusters of tumor cells. We follow a protocol that was developed by the colleagues at Penn and has now been used by a number of people. And you dissociate the cells and put them in a rotating shaker, and you're able to preserve them over time. So we treat them with a demethylating agent, a DNMT1 inhibitor, or DMSO, and then after 2 weeks, we profile them for single-cell RNA-seq. So what we observed after treatment with a demethylating agent was an increase in interferon gene expression and interferon signaling. This was seen only in the low-grade tumor. Note here that this first model is a grade 2, and this one is a grade 4, and this one already had experienced loss of the interferon locus, which is on chromosome 9. In addition, we see that there's increased expression of endogenous retroviral elements after treatment with the demethylating agent. And this is essentially what enables the increased interferon signaling, because as you know, when you have increased expression of endogenous retroviral elements, these essentially viral particles that get recognized by sensing mechanisms and lead to activation of immune response. By applying the treatment with the demethylating agent, there was increased expression of these elements and increased interferon signaling. Then we went back to that case that I just showed you in the previous slide that we had treated with the IDH inhibitor pre and post, and we also see that after treatment with an IDH inhibitor, we see increased interferon signaling, and we also observe this when we do bulk analysis on a number of other tumors. At around the same time, Nabil Bardisi, who's another colleague at MGH, published another paper where they did similar experiments and showed a similar effect of induction of interferon signaling by treatment with an IDH inhibitor. So, in summary, for this first part, by leveraging patient samples, we show that the transition from low-grade to high-grade in IDH mutant glioma is characterized by expansion of the NPC-like cell compartment, that this is associated with progressive loss of methylation and rapid cell division, and that IDH mutant gliomas are essentially epigenetically driven tumors at the onset that subsequently evolve by acquiring additional molecular alterations that become genetically driven diseases. And there's a window of opportunity early on with the treatment using IDH inhibitors or other demethylating epigenetic therapies to really try to restore the normal epigenetic state and also have an effect in terms of preventing immune suppression, which is the other mechanism I was just describing.
Hey, Nate, just a quick question. In the slide before, for the low grade,
Yes.
those organoids that you grew,
Yes.
do they take a while to grow? How? Because they're not easy to grow.
That's a very good point. Yeah, so for low-grade tumors, these are frail models, and they require a lot of patience. And also, you cannot maintain them with.
That's why you cannot attach it or,
Exactly.
Gotcha.
Yeah.
I'm trying to wrap my head around what are the ultimate consequences of this like increased astrocyte cell population. So do the IDH inhibitors, or is there any evidence that they also decrease proliferation, or is it like the tumor's still going to grow? It's just going to be the astrocytic type lineage instead of the neural progenitor cells?
Yeah, so they decrease proliferation by the fact that you're going from a state where proliferation is greater, you're shifting that to a more quiescent state. So those AC-like cells really don't proliferate at the same rate. So overall, you're increasing proliferation.
But the tumor will still grow over time.
Over time, it will. Yes. But that's why these are not curative therapies.
I think they're failing after like a year, right?
Yes.
No, well, the Indigo study that led to the approval by the FDA of this showed that compared to placebo, patients that were treated with vorasidenib had progression-free survival of 27.7 months compared to 11.1. In real-world data that we published and I'm showing here, we find that the effect is a little bit longer. In fact, this patient that I just showed you is being treated with an IDH inhibitor. I started him on ivosidenib 5 years ago when I first met him, and he's still stable. So my hope is that patients will get a lot more benefit. It'll buy us time to come up with better things to treat about this.
Sure. And then
A couple of pharmacologic questions. So with the clinical doses with these agents like vorasidenib, what percent activity is reduced in the brain? Is it 100% reduced? It seems like does a systemic toxicity override pushing the dose? And the reason I get to this is, recently I had a patient with uveal melanoma metastasized to the liver, and they did portal percutaneous hepatic circulation isolation, just hammered the liver at doses that would have destroyed the bone marrow. And that's a fairly effective technique. So, are we dose limited? What's the residual activity?
Good question. So in 2023, February of 2023, the main group doing this work, which has been led by Ingo Melnikov and Morris Terry, published the results of the surgical window opportunity study that was run in parallel, but a little bit earlier than the Indigo study. And there they compared ivosidenib, which is the initial IDH1 only inhibitor, with vorasidenib. And they show that the brain penetration of vorasidenib is about 10 times those of ivosidenib. But both compounds, interestingly enough, lead to reductions of 2-hydroxyglutarate, the ONCO metabolite, which is a good biomarker of response, to levels of about 83%. The current dose that we use for vorasidenib is 40 milligrams daily, and that's relatively well tolerated. But at higher doses, they saw a signal of hepatotoxicity. And in fact, we see hepatotoxicity with some of my patients. I just dose reduced a patient on Wednesday when I was in clinic. So I think that that's the therapeutic window that we have. But I think that with that level of inhibition of 2AG, we're still getting a significant clinical benefit.
If I understand, James, your question is there a role for us to consider doing more local therapy, just like what they did at the liver, actually? And I think that there is opportunity for us to think more thoroughly about that and hammer these tumors more aggressively, you know.
So with an epigenetic therapy, we're working, these are not cytotoxic therapy. We're not killing cells. We're just trying to restore their normal function. And the timeline for seeing a response is in the order of weeks. The actual timeline for activity of any effect in the lab. So is not really amenable this particular pathway for treatment, direct treatment or intraoperable treatment. But there are other opportunities. So IDH-mutant gliomas have multiple metabolic vulnerabilities. I'm going to tell you about another one in just a minute. But my colleague and collaborator, Dan Cahill, who's a neurosurgeon at MGH, has done some beautiful work showing that drugging of an enzyme called NAMT can lead to benefit. However, oral NAMT inhibitors are incredibly toxic. When he developed a we get these messages all the time. So he developed a NAMT inhibitor that was embedded in a dissolvable pegylated product that they applied directly to the tumor cavity. And they did a study, Ganesh Sankar's first author of PNAS a few years back.
And I think Wendy regional.
Of course, yeah.
Here's a quick kind of question or comment, Marissa, and then I'll let you continue on. But I think, partially what I'm deriving in terms of the clinical opportunity for this is really we need to use the best time to use the vorasidenib is upfront, whereas many of us, previously have been treating with radiation chemo, they recur, and then we put them on this, and we haven't seen it work. And you're showing us why it doesn't work, and that it really has to be the first treatment you do
Right off the bat.
Yeah.
Yes. Gotcha.
I'm treating, all my patients during the study. They had subtotal resections. 80% of patients had at least 2 centimeters of disease remaining going into the study. But I've even treated patients with gross total resections, clear changes, because we know that despite all the neurosurgical skill, it's never taken all out. So, right. So now I want to tell you about another project, bringing the bench to the patient. And this is based on beautiful work done by my collaborator, Diana Shi, who's in the Department of Radiation Oncology, now at MGB, and Sam McBrayer, who's a PI at UT Southwestern. He trained with Bill Kaling at Dana-Farber. And they identified that drugging of the de novo pyrimidine synthesis pathways through inhibition of dihydroorotate dehydrogenase, or DHODH, it's therapeutic vulnerability. Blocking this enzyme leads to a mismatch in terms of nucleotides, and this leads to increased cell death. And this was validated in a number of patient-derived models and a beautiful genetically engineered mouse that Diana Shi developed. And I think it's one of the most exciting animal models that we have of the disease. And then on patient-derived xenografts, we see that there's a survival benefit treating with this DHODH inhibitor, and we see that through this mechanism, through inhibition of DHODH, which leads to a significant decrease in the ratio of orotate to carboxyl aspartate, which are the enzymes that are proximal distal to the the products that are proximal distal to the enzyme. So, in light of their discovery, we partner under the auspice of our U19 glioblastoma U19 grant. This is a large grant that we have at Harvard and Stanford and collaborate directly. And I'm involved with Project One, where we're designing a surgical window opportunity study to evaluate dihydroorotate dehydrogenase inhibitor in patients with WHO grade 4 IDH mutant astrocytoma. When the grant was posted in 2019, it was before the 2021 classification, so there was still such a thing as IDH mutant glioblastoma. So that's how we were able to get this project into the grant, and it continues. So here, patients are going to be treated with the dihydroorotate dehydrogenase inhibitor. And then they're going to go for surgery. We're going to do a Mars spectroscopy also to assess response before the operation. They go for surgery, and we have a number of experiments planned on those samples. So that irrespective of what they learn in terms of clinical efficacy, we're going to learn something about the biology of the disease and the mechanistic response to this treatment. And more recently, under the auspice also of our spore grant, brain spore grant that we have with Stanford, I'm involved with Project 3, which is looking at treating patients with IDH inhibitors that are low grade, treated with an IDH inhibitor and have failed. Those patients that have 3 years or 4 years show evidence of progressive changes on their MRI. We're going to treat them with this dihydroorotate dehydrogenase inhibitor and evaluate response to treatment. But in addition, we're going to have an arm where we're going to look at mechanisms of resistance to the IDH inhibitor as well. And we have a number of experiments lined up with the surgical samples. Let me transition now to glioblastoma. And this is also a patient. He was 74 when he presented to me with and I like to show this slide. T.S. Noodle was a neuropathology fellow at MGH and trained on the K-12 grant, and I now co-direct. And he published this in 2011, really highlighting the invasive and heterogeneous nature of glioblastoma. And here, using probes that are specific for PDGFRA and EGFR, he shows that in different areas of the brain that have been affected by the tumor, these two particular genetic drivers are expressed at different levels. And this is a question that we had for a long time. Is glioblastoma one tumor or many? And with the genomic revolution and the initial characterization of glioblastoma, Roel Verhaak, who is a pioneer in this field and a close collaborator, in 2010 published this paper where they did bulk RNA-seq on glioblastoma, and he essentially established that there might be 4 types of glioblastoma: one that is proneural, one that is neural, and one that is mesenchymal, and a classical one. So essentially, when you had glioblastoma, apparently you had one of those 4. But then shortly after, my mentor, Mario Suba, published a study, one of the first single-cell RNA-seq studies in glioblastoma. And this is a study that has about 400 cells. I mentioned that because this was a very laborious process back in the day to be able to do this. But he shows that within tumors, there are cells that behave like proneural, neural, classical, and mesenchymal. So this starts to shift the paradigm from thinking that there are 4 different types of glioblastoma to showing that, in fact, there are many different cell states within glioblastoma. And this leads to the landmark paper that I was very fortunate to be a part of back when I was arrested. I rotated in Mario's lab in late residency, where we show that the cellular heterogeneity of glioblastoma is characterized by 4 main cellular states. And more recently, through collaborations with Alyssa Greenwald and Tai Tirosh, we leverage spatial transcriptomic methods to really try to explain where in space all these different cell states are. And we see that necrosis and hypoxia is an organizing principle. And out of that, we have a gradient followed by more mesenchymal cells, and then further out in more oxygen-rich areas of the tumor, the neural progenitors. And then earlier this year, through a large collaborative project led by Masashi Nomura, Avishai Spitzer, we reclassified by single-cell RNA-seq a large number of glioblastomas pre and post-treatment, and we define additional cellular states that are smaller, but we are not able to resolve them. Well, before we didn't have that resolution, and this includes the GPC cellular state that we described on IDH mutant glioma, as well as some neuronal type of cellular states that might be the ones that are related to the input, the neuronal input that tumors have. And then we showed that both at initial treatment and at recurrence, the cellular state composition is not that different. There's some changes in terms of number of malignant cell fraction and purity of the tumor, but as I was alluding earlier, Dr. Quinones, this is probably related to the sampling that was done. This was using a technique called nuc-seq, where we do nuclear RNA-seq. The 2014 and 2019 papers were using a technique called SMART-seq2, where we dissociated tumors and we plated them. And we had more depth, but we probably lost some cell types and some cell states through the dissociation process and the sorting. So one of the important things to know, and this is also in the 2019 paper, a beautiful set of experiments done by Toshiro Hara, is that if you take, you can identify a specific cellular state by looking at a cell surface marker. So CD44 is expressed mainly on MES-like cells. And if you deplete every cell by the CD44 and then you plant those in a mouse, in a nude mouse, and over time you sample that again, you see that the tumor is able to reconstitute the heterogeneity of the initial tumor. And this is driven by mechanisms that are coming from the environment. This is another study led by Toshiro Hara, where expression of oncostatin M was associated with driving the mesenchymal state. And more recently, there's a lot of enthusiasm about other cytokines, as well as neuronal input into the tumor driving specific cellular states, or likely the stem-like states. And then, in addition to that, since this was done in a nude mouse, with no strong immune microenvironment, we were also interested in looking at cell intrinsic mechanisms. And then we had these two aims. One was to characterize chromatin accessibility and gene expression concurrently in GVM, and then to identify regulatory elements associated with each cellular state and experimentally evaluate their potential to enable cell state transitions, essentially plasticity, all done in patient-derived models. So this is the schema here. We collected a number of samples and we obtained a number of samples from other collaborators and profiled those by ATAC-seq and RNA-seq. We defined cis-regulatory element modules associated with each cellular state. We constructed a gene regulation network, and we're in the process now of doing experimental validation. So just to make the point, in that paper from Patel and colleagues at Mario Sua published in 2014, we had about 400 cells. Now we are profiling cells in the order of thousands of cells. And this gives us a lot more power to make observations. And, we cluster by cell type. We always do the sanity check of making sure that these cells are true malignant cells, and we do this by inferring copy number alterations. And as we would expect in glioblastoma, we see gain of chromosome 7, loss of chromosome 10. And we went on to use non-negative matrix factorization to define these cis-regulatory element modules. And we defined 6 of them, 2 of them that are universal and are shared among the different cell states. But then we identified 4 that are cell state specific. And looking then at the transcriptional identity of those cells, because we have multi-omic data, we're able to put this plot here, which I think of it essentially as a Rosetta Stone of cell state plasticity in glioblastoma, that tells us what cell states are more likely to transition into other cell states and identify pathways to be able to modulate and control that. And we plan to do that because we have identified rich motifs within each of these cis-regulatory modules. And you see, for example, for the mesenchymal-like, we see a lot of AP1 transcription factors and then a lot of neurodevelopmental transcription factors for the MPC-like. And interestingly, we see the SOX family of genes really cutting across all of them. We're studying SOX genes as potential master regulators of plasticity in glioblastoma. And this is a platform where we're overexpressing different transcription factors and seeing the effect on cell state distribution, while at the same time, we're knocking down through perturb-seq experiments and seeing if we see the opposite effect. So in summary, malignant cell states in glioblastoma can transition between each other and maintain intertumoral heterogeneity. Cellular state plasticity is facilitated by interaction with the tumor microenvironment and enabled by epithelial mechanism. And concurrent profiling of gene expression and chromatin stability in glioblastoma reveals that there are unique cis-regulatory modules associated with these specific cellular states, and enriched TFs within each cell state. Specific cis-regulatory element modules can be overexpressed or repressed to enable transitions between cellular states. I think this will lead over a period of time to our ability to really control the heterogeneity of the tumor and leave the tumor vulnerable to therapies. In the meantime, patients need treatments. So I've been involved in helping advance other efforts. And I want to tell you briefly about this phase 2 multi-center clinical trial that I'm leading, where we're evaluating the combination of a PARP inhibitor, olaparib, with pembrolizumab, and temozolomide. And this is a study that we're running out of Mass General Brigham, Dana-Farber, of course, Memorial Sloan Kettering, and Israel Alehi. And here, again, the surgical window opportunity model, I think that should be the model that trials should follow moving forward, so that we can learn something from every study. Patients are being treated with a triple combination of pembrolizumab, olaparib, and temozolomide, or with pembrolizumab alone. Then they go for surgery, and then we retrieve those tissues for analysis. We know from the Checkmate study that single-agent immune checkpoint inhibitor does not lead to clinical benefit in patients with glioblastoma. And the hypothesis here is that using the PARP inhibitor and increasing double-stranded DNA damage, which has been shown preclinically to lead to increased interference signaling, might synergize with pembrolizumab and lead to increased immune activation and anti-tumor immunity. So here, of course, we have a clinical endpoint. We're looking at PFS6, but we're also looking at tumor-infiltrating lymphocytes, correlating that with response, looking at malignant cell signatures and interferon gamma signatures. And this is all work that I'm doing in collaboration with Will Prince at the University of California in Los Angeles. And lastly, this is another project that I'm very excited about. This is leveraging a technology developed by Oliver Jonas, one of our scientists at the Brigham and Women's Hospital, and first evaluated by my neurosurgery colleague, Paolo Peruzzi, using a microfabricated device that is just 6 millimeters, is above the tip of a pencil, and has 27 wells that can be loaded with different compounds. So you can load 9 compounds in triplicate, and then that can be inserted into the tumor. And then the device and a core of tumor around it is extracted, and you can quantify response to therapy in vivo in the patient, personalized. And this was validated in a 3-hour experiment. So patients have the device implanted, 3 hours went by, and then it was removed, and the section was completed. And more recently, we completed a 72-hour experiment where patients come in, they get the device implanted, they leave the operating room, they go back to the floor, and 3 days later are brought back in to remove the micro device. And I think this gives us better understanding of the response to therapy. And one of the things that was noted in that study was that a traditional predictor of response to alkylating chemotherapy is the presence of methylation of the MGMT promoter, the 06-methyl-methyltransferase. But it's not a perfect predictor because we all have had the experience of treating a patient that was unmethylated and not expected to benefit and seeing that they did very well. And at the same time, we treat a patient that is methylated and expected to benefit and does not. And this was the case here. As you can see, this patient was unmethylated, didn't benefit as expected, but this patient was methylated and did not benefit. And this patient that was partially methylated, which those tend to align more with the unmethylated patients, benefited the most. In the assay and clinic, and I'm not showing you the clinical data here. So using this, we are now designing and trying to secure funding for a very important aspect of translational research. A prospective study where we will enroll newly diagnosed or recurrent patients with GBM. The patients will have the micro device implanted. We'll retrieve it after 72 hours, and we will offer them temozolomide or CCNU. And then at progression, we're going to be evaluating, we will only need 6 wells of the whole device to evaluate response to these 2 agents. We're going to be evaluating other therapies that, if we can make them available to them, would be therapies that they could receive after progression with the standard therapies. But at least this will give us an opportunity to really be more specific in terms of predicting response, helping patients that will benefit the most and avoiding the toxicity. These drugs, particularly CCNU, have incredible myelosuppressive activity. Sparing those patients that will benefit. Everything.
That looks really great. I had a question. So you leave the device for 3 to 72 hours, and how long does it take to measure if the medication works or not?
Yeah, so then the device and the core of tissue around it is taken out, is sectioned, and then our colleague Oliver Jonas runs a number of different assays, including phosphorylated gamma H2X, to look at DNA damage to try to predict response. So I finish up by saying that over the last 2 decades, scientific advances have enabled us to ask questions of increasing complexity about the biology of gliomas. We obtain the most accurate and relevant answers to these questions when we interrogate tumors directly, either obtaining tissue from patients or by doing experiments in vivo, like the one I just showed you before. And surgical window of opportunities trials offer a unique chance to never have a negative trial again, because we're going to learn something from every trial. Nelson Mandela used to say, "I never fail because when I win, and when I fail, I learn." And here, when we fail, we're going to learn. And we're going to move forward. And we owe that to our patients. But we have to be mindful that as we do studies with patients, increasingly more complex technologies, we need to be laser focused on making sure that we ensure their safety, and whatever science we do, our center objective or single-minded focus is on their best interest. Or as someone dear to this house says, the best interest of the patient is the only interest to be considered. One of the great pleasures of doing this work is getting to know and work with and learn from a number of different collaborators. I would like to particularly acknowledge Mario Suva, who has been an incredible friend and mentor over many years now, as well as a number of other collaborators at my institution and at other institutions here and abroad. My funding sources and above all, my patients and their families. So I'm happy to take all questions.
Comment, it's interesting what you presented with the device. I don't know if you use the Kaya panel ever, but you take the tumor out and send it to them, and they do a drug panel, and they found similar to what it looks like you found is that their prediction of temozolomide responsiveness did not correlate with MGMT, and so I think that just proves how important something like your trial with the device is. And I guess the question I would pose is, as we're coming up with drug panels to try to, test responsiveness to align with treatment, is there any testing we want to do beforehand, such as a biopsy to look at, the complexities of the tumor, or eventually a blood test, so that then you can really personalize what panel you're sending. For example, you're not sending an EGFR drug on someone who has no EGFR alteration.
No, I think that's a great idea. And I think, there are multiple lines of work in this regard. I haven't used that specific assay, but at the Alliance for Neuro-Oncology meeting last week, there's a concept that was presented, it's been presented a few times, where they're looking, I think it's called chemo ID. It's take a tumor sample, send it to a company, runs screening for a number of different compounds, trying to do personalized systemic therapy. And yeah, I think that all these things need to be optimized. How much information we can gather upfront I think it's gonna increase. I'm excited, as we were discussing last night at dinner, about the possibility of running some molecular characterization assays on CSF studies. I think with the amount of information we're going to be able to extract just from imaging, there are very good machine learning classifiers that will tell you the presence of an IDH mutation just by looking at MRI data, by know about molecular restrictions. Yes.
That's a fantastic talk, and thanks for
We're
coming.
gonna be meeting Ramsey today. We just recruited him.
So really excited to be here. A few questions, like I think from what you're presenting on all your work, very elegantly done. And there's been a lot of work in other tumor types as well, like in terms of tumor plasticity and how we could potentially kind of really constrain tumor plasticity for clinical benefit. And of course, one major class of drugs that has never realized its potential is DNMT1 inhibitors, right? So like all cytidine, 5-ASA, we've done like numerous trials in several tumor types. Unfortunately, in a clinical setting, it never panned out. But it's really fascinating opportunity given your device, and specifically in GBM, if you can kind of like really investigate these drugs with the correct pharmacology. Because the reason these drugs never worked is because of the terrible pharmacology and how those trials were. The NMT1 inhibitors are rapidly deaminated. It's like CDA. They've been like some work, but anyway, they've not really been used as epigenetic drugs because the dosing is so high that it's causing cytotoxic effect rather than the pure epigenetic effect that we desire, right? So I think there's a unique opportunity to kind of really optimize the pharmacology of these agents, especially for GBM. I think.
Yeah, thank you for making that comment. I have a great clinical and research interest in IDH. And I like this patient because a lot of them are my age, and they have young kids that are struggling with the same things that I struggle. On top of that, they're struggling with their brain tumor. But I have been talking to Pier Paolo Peruzzi about having an including IDH patients trying demethylating agents, such as DNMT1 inhibitors. It's a narrow therapeutic window, as you alluded to, but at least studying them with setting. In fact, I was talking to a representative from the company because I think there's a possibility of synergism between the IDH inhibitor and demethylating agents in these patients. And I think they're looking to do a small study. The other thing with this disease is that when you start treating the low grade phase, the clinical endpoints take a long time if this patient might not progress for many years. Companies sometimes, fortunately, don't have the patience to do some other studies. So studying in the setting might give an early answer that might entice them then to.
And that's exactly the setting, and that's the population where the epigenetic inhibitors, especially DNAP1, you need like adequate number of cell exposure in this phase, right? The low-grade tumors are the ones that are the challenge.
Exactly. So there's the logistical and the scientific barriers that we're trying to.
Can you go back a couple of slides to the device, Science Foundational Medicine, if you don't mind? This one right here, and the drug susceptibilities, because now we think about it, and some of us do think about the limitation and the reason why these trials are difficult to find. Quick question: who owns the IP for this device?
I think it's owned by
The people.
the developer, Dr. Jonas, and he holds a patent at the hospital, of course. Yeah.
And that's really the places like us, Mayor, can tell you that we can invest a lot of resources. What we're asking ourselves is what is the return on investment for us as an institution? Of course, we want to help the patient first and foremost, but then to be able to help the patient and run the phase 2, the phase 1 of the trial, we estimate for a brain tumor between $5 to $7 million. The federal government doesn't have that money. Very few institutions have that money. We haven't had that money. The problem is that then you go to the phase 2. That is $10 to $14 million, phase 2. So who's going to put up that money? So what we're trying to think for ourselves is how do we create a value in the work that we're going to do in such a way that we will have no problem raising that money for the phase 2. Otherwise, the program is just going to end up in phase 0 and really not go to the next level unless the company or someone else takes it, and then you work for someone else basically. I'm asking our teams, our members, to always think about how do we create value for ourselves? How do we put IP in our ideas? How do we create devices? How do we partner with biomedical engineering? So that way the IP is within the institution, within the team, and then it makes it much easier for you to either do a startup, which are the things that we're promoting with our faculty, or license the technology, but the work is done back at the institution. We're just doing right now with epilepsy, with a major grant that we funded that is in epilepsy for all 3 sites, but the IP belongs to Mayo. We license the technology to the company, but we're going to do all the trials at Mayo. And therefore, as the company grows, the return on investment for us to be able to invest more is there as well.
I think that's a great model, and I think that's a model that we see, successfully implemented and replicated in Boston. That has a very vibrant biomedical ecosystem, a very vibrant venture capital in biotech, and we cannot do everything on our own, and we need to partner up with other folks and try to align interests and goals. Us looking for the patients, them looking to generate a return, but I think that there are ways to work together.
It's great. This is amazing work. Having you speak our language, we're doing a lot more. I think that you're going to be meeting with Nancy. You met with James already. Going to be meeting with us. We are, and probably Wendy told you, we're doing a lot of stuff. Window of opportunity, surgical, and many idiomas. We're doing the same. That's what Craig is not here. He's been doing a lot for local therapies. So we have a window of opportunity in the operating room as well, and we don't want to break it all. Not only for diagnosis, potential prognosis, but just therapy itself, that we can take advantage of for all different tumors. So we're very excited. Thank you, Nick. Wonderful talk. Any questions online? Say yes, please. I'm sorry, Emiliano, please. I know that we're a little bit of time, but please.
Dr. Mead, a lot of congratulations. And she asks, what do you think is the tumor microenvironment stimulus that creates all these epigenetic changes you described? And 2, do you think this plasticity is different at a regional level within the tumor?
Yeah, great questions. I don't know for a fact that the tumor microenvironments directly leads to the emergence of the epigenetic mechanisms. I tend to think of those being separate, interacting but separate. And you know, I think we're starting to learn a lot more. I didn't get a chance to look into it, but I recently was able to partner with my colleague Tyler Miller, who's now at Case Western, published earlier this year, a really in-depth profiling of myeloid cells in the tumor microenvironment. We see different myeloid populations having different roles. So I think there's a lot more that we're going to be learning in the coming years about the tumor microenvironment input. And then, yes, I think that as this is both a function of internal epigenetic mechanisms and tumor microenvironment, as the environmental milieu changes in regional areas, more hypoxic, less hypoxic, more neuronal input, less neuronal input. I think that there's a differential there. We're still not able to capture that with the resolution of instruments that we have, that we're going to be able to do that in the future.
Thank you very much. Those were all the questions. Chat.
Thank you, Nick. Thanks, everyone. Have a great day. Thank you.